Thank you. So I'm going to talk to you a bit tonight about a virus that we work on, and it's this virus that actually drives the kinds of lymphoma that Lee suffered from and that a lot of people across the world suffer from. And the virus is a virus called Epstein Barr Virus. And this virus was actually the first virus to be associated with a human cancer. It was isolated in the 1960s in cells that were grown from a cancer sample taken from a child in Africa. And actually, the Burkitts lymphoma, one of the key cancers that Epstein Barr Virus, or EBV as I'm going to call it, causes, still accounts for about half of all the childhood cancer cases. So it's still a big killer in Africa, and it's still a big killer across the world. And as well as causing Burkitts lymphoma, it's also associated with a number of other lymphomas and other types of cancer. And they include Hodgkin's lymphoma, the kinds of lymphomas that you might get post-transplant if you're taking immunosuppressive drugs. Those particular types of cancers particularly affect children. And also with another type of lymphoma called diffuse large B-cell lymphoma. And you probably can't see, for those of you who can, at the bottom here there are a couple of pictures of what the virus looks like. So on the left we have an image of the virus taken with an electron microscope, where you can see actually through into the virus, and in the centre there you can see the genetic material of the virus, and that's docked onto the outside of a cell that it's infecting. And on the right you can see a surface view of the virus. So this is what the surface of the virus looks like, and there are molecules on the outside of the virus that allow it to dock onto the cells it infects. So how does this virus cause lymphoma? Well, what the virus does is it infects a certain type of blood cell, the white blood cells, circulating around in your blood, called B cells. And the reason it's so deadly is because it has the capacity to drive these cells to grow continuously. So your white blood cells normally only live, and this cartoon at the bottom here is just showing you red blood cells, and the kind of lumpy green things are the white blood cells circulating around. Now normally those white blood cells only have a life of about 2 or 3 weeks. But when they're infected with the virus drives those cells to grow continuously and out of control. And that's a key feature of all cancer cells, and that's why it has these cancer-promoting properties. But actually, the conundrum about EBV is that actually most of us in this room are infected with the virus, and we don't all get EBV-associated cancers. And that's because we know that we have a really important control mechanism in us to control the virus infection, and that's our immune system. So long as we stay healthy, generally we can keep the virus-infected cells in check, and our immune system destroys them, and we live in a balance for life. But actually, if something disrupts this balance, so that can be the situation I've explained to you, where if you have a transplant and your immune system is knocked down, you're more at risk. The virus-infected cells can get a hold and take over and might grow into a lymphoma. Or if other events happen in the cancer cells, and that sounds very general, but there are a lot of other events that we don't understand, a lot of genetic changes that can happen as a result of environmental factors, and other chance factors that might happen to an individual that combined with a virus can then lead to those cells becoming cancerous, such that in a few individuals the virus is able to drive cancer development. So what do we do in the lab? That's a little bit of background that I hope explains a bit more about the science behind why we're working on this virus. So we want to find out how EBV turns one of those normal B cells into a cancer cell. And what we know is that when EBV infects those cells, it produces proteins, virus proteins in the cells, and those proteins make a difference as to what genes are turned off and what genes are turned on in those cells. And that's the key to how it's driving cancer development. So we know that which genes are switched on and which genes are switched off in every cell in the body is really important in controlling the growth of that cell and whether that cell dies or not. So the genes that are switched on will make it grow in appropriate circumstances, or they can actually trigger cell death in some circumstances. And what EBV does is it overrides the normal controls in the cell, and it turns genes on that push the cells to grow, and it turns genes off that would cause the cell to die. So it completely reprograms and changes all of the control mechanisms that are going on in the B cells. What we're studying is how the virus does this. I'm a biochemist by training. We use a lot of biochemical and molecular techniques in the lab. We also grow the cells that have come from patients and that we've also artificially generated in the lab. And it's only by understanding how this happens and how the virus does this that we can then design drugs to stop it happening. So Bloodwise is a really important charity in funding what we call basic research, which is research into the mechanisms of exactly how things are going wrong, so that that can then feed into drug design. And that's really important that we keep doing that, because the distance between what we call the bench, where the experiments are happening, and the clinic is getting closer and closer these days because of the advent of such great new modern technologies. But we have to base our new therapies on the underlying science that we need to still understand. So this just shows you some pictures of us doing experiments in the lab. So on the left here are actually some Burkitts lymphoma cells, and we can grow these in an incubator at body temperature at 37 degrees, giving them appropriate nutrients, and we can do experiments on them to figure out how they're growing and what's going on in them. In order to keep these cells growing and to keep them sterile, we have to do any manipulations that we do with them, giving them fresh food, in a special flow hood. And this is one of my PhD students here who's feeding cells in a flow hood. And what else we do is we work with bacteria in the lab. We use those as a tool to grow up pieces of viral DNA. And this shows another one of the students in my lab working with an agar plate with some bacteria that contains bits of viral DNA in it. And then we sometimes put these bits of viral DNA in the cell to try and figure out which bits of the virus are overtaking the cell. And we then take the cells and we extract proteins and DNA from them. And you can see in these tubes on the right here, one of my researchers has done an extraction and is carrying out an experiment with them. And yes, we really do use multicoloured lids and we do wear purple gloves. This isn't just for effect. And then the really difficult thing comes. We have to try and figure out what it all means. So we might come up with a hypothesis, we might do some experiments to test it, and then we might get results that we really didn't expect. And this is me with a couple of my researchers trying to figure out what on earth that result that we're looking at means, so that we can design the next experiment and move forward and find out more. And that's all I really want to say today. As I said, if anybody wants to come and find me and find out more about what we do, please do. This is a number of my lab members who would all like to say a huge thank you for all the fundraising that you guys have done through the Live It for Lee campaign. So a huge, huge thank you from us for all your amazing fundraising. Thank you very much.